Morning, everyone. In this SARANSH PhD thesis competition, the title of my presentation is Understanding the Regulatory Role of Sugars and Their Dynamics on Flowering in Saffron. Saffron is a triploid with flowering totally dependent on temperature. From May to June, there is floral induction, and from July to November, there is floral differentiation and development. This is a comb. This is a comb. It is placed at ambient to high temperature without soil, without nutrients and other resource materials, and still it develops into the whole plant. It means that there are stored metabolites inside the comb which trigger this comb to develop into the whole plant. That is the power of the storage of saffron comb. A comb analysis found that 60% is mainly carbohydrates. To investigate the role of temperature in saffron flowering, we designed an experiment and put flowering competent combs at two different temperatures: ambient high temperature 25 and low temperature. Ambient high temperature promotes while low temperature suppresses flowering. After we did the transcriptomic analysis of the apical bud, we found that starch and sucrose degrading genes were upregulated at ambient to high temperature, while raffinose phosphate genes were upregulated at 8 degrees Celsius. It means that sugar metabolism plays an important role in temperature dependent flowering in saffron. At sugar metabolite level, starch was downregulated at 25 degrees Celsius, and raffinose was upregulated at 8 degrees Celsius, which leads to the suppression of flowering, and other sugar metabolites, especially trehalose 6-phosphate, were also differentially accumulated. Exogenous treatment to confer sucrose induces while raffinose suppresses flowering induction. This is the overview of the trehalose 6-phosphate and raffinose signaling in saffron. Heat map in this pathway shows that trehalose and raffinose phosphate genes were differentially regulated. Expression analysis showed that the TPS gene, which is induces while gloss suppresses floral induction, as their expression was differentially regulated during the floral induction time. The two-bit expression of CSTPS, it is the trehalose phosphate synthetic gene, resulted in early flowering, while via Wiggs leads to the flowering suppression in saffron. This is the expected outcome model on how sucrose, raffinose, and trehalose 6-phosphate ratio can influence the temperature-dependent flowering in saffron. Finally, I would like to acknowledge the CSIR, IHBT, CSIR, Indian National Science Academy, and ACSI for giving me the opportunity to present my work. Thank you so much.